the polypeptide chain is sort of stretched out and being shown schematically. So basically, a septin has three regions to it, what we call domains: an N-terminal domain, which includes what is called a polybasic region. I'll come back to that in a minute. In other words, it's a region full of basic positively charged residues, arginines and lysines. It has a central domain, which binds the small molecule called GTP and can hydrolyze it to produce GDP. And it has a C-terminal domain, a third domain, here shown in black, which I will also be talking about a bit more in the future. Well, it turns out that the human genome codes for 13 different septins, and they all get numbers: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 13 is missing because we're superstitious, and then there's a septin 14. Actually, it's not because we're superstitious, it's because septin 13 has been discovered to be a pseudogene. So if you're hearing about septins for the first time, it will be very difficult to follow all of these numbers. So I'll try and make things easier for everyone by using colors instead of numbers. So these 13 septins can be divided into 4 groups based on how similar they are. Similar in terms of amino acid sequence. So there's a group which I will refer to as the green septins. There's 3 of them. There are 5 blue septins. There are 4 red septins and 1 yellow septin. I apologize. My cell phone has decided to go off. I apologize. So I will refer to these 4 different groups by their colors. And in order to form a filament, it is necessary that you combine, and not in any old way, but you combine septins from these 4 different groups. So in order to form a filament, you can either do that using 3 different septins, in which case you need a yellow, blue, and a red, or 4 different septins, in which case you include a green septin as well. And these different filaments are based on core particles, what we call core particles. If it's based on 3 different septins, then the core particle is actually a hexamer. So it has 2 copies of red, 2 blue, and 2 yellow. If it's based on 4 different septins, then it forms an octamer, and you have 2 copies of each one of the different colored septins. Okay, now, what's happened here? Oh, apologize. Okay. For some reason it was slowing. So this is just a summary then. It looks a bit like stamp collecting. This is a summary of all the different structures we now have for individual septins. And not looking at a filament, but an individual septin. It turns out that crystallizing these oligomers, these hexamers and octamers, is very difficult. We've been very unsuccessful in doing that. But crystallizing individual septins turns out to be much easier. And we've now collected a whole load of structures from each one of the different groups and from different species. So this is what is currently present in the PDB, the protein data bank, all the structures that we know of. Now, if I were to eliminate now all of the structures which have been solved in the developing world, in a particular case in Brazil, you will see the impact that we have had. So all that is left is what's been done by the so-called developed world, the first world. So it's a sign that it is possible, by choosing a particular niche area, that it is possible, even in a developing country, to make a significant impact to a particular field. And I'll try and convince you that this is not just a matter of choosing an uninteresting subject, which nobody in the first world cares about. It's actually very important. The structures of septins are very relevant for several different things, and one of the examples I'll give you in a minute is Zika virus. Okay, so what we do is we take septin crystals, we can take them to our old local synchrotron, the UVX machine. These crystals, which is septin 3, it's a green septin, only diffract about, well, worse than 6 angstroms, but if we take those same crystals to the SRF, then with a very even smaller crystal, we can get data to 2.9 angstroms, and we now have structures at 1.7 angstroms. So access to big infrastructure, to facilities all around the world, has been absolutely fundamental for doing the kind of work that we do. So we don't really care where we have to go or where we have to send our samples. We'll often do this by remote data collection back here in São Carlos. but the samples can be anywhere around the world. So we've used the SRF, as I mentioned, Soleil also in France, Alba in Spain, and most of all Diamond in the UK, in order to keep ourselves at the cutting edge of septin research. And now, as I mentioned already, we have our new shiny synchrotron, the Sirius machine, which we've collected our first data on at the end of last year. Then it had to close down to have the robot fitted. So, as I say, the characteristics of this machine in terms of emittance and energy, etc., are very similar to Max4, which is probably well known to most. Okay, so getting back to septins then. Yes, so we've been successful in solving individual septins, but as I pointed out at the beginning, septins don't act on their own. They act in the form of these heterofilaments. And here we have the only crystal structure solved at 4 angstroms resolutions in 2007. It's the only crystal structure available to this day of a complex where we have 3 different septins involved. So we have a red, a blue, and a yellow. Okay, and over here on the left, you see, as I said, what is called the core particle. It's actually a hexamer. So this is formed of 2 reds, 2 blues, and 2 yellows. But there's an ambiguity about what the structure of this hexamer would be, because in the asymmetric unit of the crystal, you have 1 red septin, 1 blue, and 1 yellow. The structure clearly establishes that that is the sequence. But in order to form the hexamer from the trimere, you must apply crystallographic twofolds, and there are 2 possibilities. You could either produce this hexamer here shown with this red arrow, or this one here shown with the yellow arrow. So the one with the red arrow leaves a red septin at the end of the hexamer, and the yellow arrow leaves a yellow septin at the end of the hexamer. So you have these 2 possibilities. And it was established in 2007 that, in fact, this is the correct answer. So it's the yellow septins which are on the ends. Now, obviously, if you think about any particular septin along the filament, it has 2 neighbors, and it therefore forms 2 different kinds of interface. And these interfaces have names. They're called G and NC. So, for example, this blue septin here forms an NC interface with yellow, and it forms a G interface with red. And I'll come back to that shortly. In order to form a filament, then, once you have established that this is the hexamer, it's just a matter of stringing them together, and that's how you form a single septin filament. Now, it turns out that we were interested in asking a particular question, and that question was related to something called Kinoshitas Rule. And Kinoshitas Rule says that if you take a hexamer, for example, and you substitute one of the septins, for example, this septin 2, which is a member of the red group, and you substitute by another red septin, septin 5, then that should be possible. What you can't do is substitute one by a different color. So I can't exchange a red one with a blue one, but I should be able, at the same position along the hexamer, to exchange any red septin for another one. So we said, well, let's take the original combination, which is 2, 6, and 5, and let's change 2, 6, and 7, and let's change 2 and put 5 in its place. And we should get the same hexamer as was seen in the 2007 study by Wittenhofer's group. And the way to prove that is you put a marker protein, doesn't really matter what it is, you put the marker protein onto one of the septins. In this case, we put it onto the red septin, which should be at the center of the hexamer. And then we make this thing electron microscopy. And what you should see then is that this marker protein called MVP really ought to this marker protein MVP really ought to be located at the center of the hexamer. But you can see it's dancing around the end of the particle. So these, we're just flipping through different class averages and seeing where the marker protein appears. So that seems to suggest that, in fact, the red septin is not at the center, it's on the end of the hexamer. And this, of course, was very heterodox sort of way of thinking, because it had been established for 15 years or so that it's the yellow septin which is at the end of the hexamer. So it could be that Kinoshitas Rule isn't true, and that the reds don't substitute at the same position, or it could be that the hexamer is actually wrong and it's inside out. So in order to convince people of that, we had to do the same experiment going back to septin 2, putting the original red septin back in position, And it should look like this, but if we are right, it will look like this. And when we do the same experiment here, it's not a movie, you can just see different stationary class averages. And the extra density is coupled to the end of the septin particle at the end of the hexamer and not at the center. So we have now established, it seems, that the hexamer is inside out. And this was not that easy to get published in the literature. I mean, we did finally get it published, but it was a little bit revolutionary. And I think there's a kind of lesson there, because once something is published in the literature, particularly if it's published in Nature, which is the case, it's very difficult to correct it if it's wrong. There is a sort of truth, as it were, which comes from the longevity of an article. The longer it's been in the literature and people keep repeating and repeating and talking about it all the time, it gets harder and harder to convince people that it's actually wrong. I sometimes stay awake at night wondering how much there is in the scientific literature, which has never really been validated and may actually be wrong. Anyway, it seems now that we have a new model for what the septin hexamer looks like. It's got the red septins on the end, and what we know now is that if we make an octamer from that hexamer, it's not a matter of adding the green septins to the end of the hexamer, they actually are inserted in the middle. And what that means is that both the hexamer and the octamer expose the red septin with its NC interface at the edge, at the end of the termini of both of the particles. And what that means is that the hexamers and the octamers should be able to polymerize into mixed filaments. Okay, so as I say, this took us a little bit of time and effort to actually convince people that we were right, but more than being right, why is it important? What's the relevance of it? Why is it necessary to know what the structure of the hexamer is? And what are its implications, and particularly what's its relevance to scientists working in a developing country? Well, in Brazil in 2015 and 16, we had an epidemic. It didn't get to the point of being a pandemic, but it's caused by the Zika virus, transmitted by the same mosquito which transmits yellow fever, dengue, etc. I'm not sure how big this was in Africa, but certainly in South America, and particularly in Brazil, it was a very big event. And it was very quickly recognized that there was a relationship between microcephaly in newborn infants and pregnant women who'd been infected with the Zika virus, but no one knew much about the mechanism behind this until 2019, when a group in Canada discovered that the Zika virus has a protease. It's called NS2B3, doesn't matter what its name is. It has a protease which is necessary for processing what's called the viral precursor protein. And it chops it up into the individual proteins, which will then form new virus particles. Well, it turns out that one human protein has the right amino acid sequence to be recognized by this viral protease. So intracellularly, although the protease is there just for the purposes of reproducing the virus itself, it has an off-target effect and actually cleaves one human protein. And it turns out that that human protein is septin 2. It's the red septin, which is at the end of the hexamer. And when it does the cleavage, when the protease from the virus cleaves the human protein, it removes the C-terminal domain, that last final bit on the end of the structure. So we decided, well, let's do some experiments to see what the impact of removing that C-terminal part of the red septin is. Well, because the red septin we now know is at the end of the termini of the hexamer, what that means is that this region could have a potential role in stabilizing the filament when the hexamers polymerize into a fully grown structure. So if we take the wild type, in other words, the normal septins, which have the C-terminal intact, and we look at those intracellularly with a marker protein, we can see green filaments being formed here. And if we look at them with negative stain in the microscope, we can see bundles of filaments being formed here. But if we remove that part of the protein from the red septin, what seems to happen is that that septin is no longer able to polymerize. So all you see are these are little hexamers inside these oval shapes here. And intracellularly, there are no filaments formed. You just get diffuse green pattern. So what that seems to suggest is that by cleaving that particular region of the red septin, the filaments and bundles of septins are disrupted. This would, since I mentioned right at the beginning that septins are important for cytokinesis, in other words, for cell division, this would lead to a delayed cytokinesis and eventually cell death, decreased proliferation, and microcephaly. So what that suggests to us is that if this is a hexamer and this is another hexamer, and the two red septins at the end of each hexamer have to come together to form a stable interface in order to form a filament, then we should look more in detail at this particular interface. That's now all of a sudden a very interesting interface to look at. And as I say, it's an interface where we have the C-terminal domain, these little red stumps coming off here, which come together. Now, if you look at the amino acid sequence of this region, we already knew that this should form an alpha helix and that these two alpha helices, one from each red septin, should pair to form something called a coiled coil, which is a dimer of alpha helices. And this should be a parallel coiled coil. In other words, the two alpha helices should be going in the same direction. At least that was the view at the time. So what we did was we took the sequence of the amino acid sequence, which corresponds just to this region, just to the little stumpy helical region, for all of the four members of the red group, and started solving their structures. So this is septin 4. And so here you can see on the left the two helices, the green one and a blue one coming from the two different subunits. And the first thing which calls your attention about this structure is that it's anti-parallel. The helices actually run in the opposite direction to one another, and that was not what was expected. Not only that, the interface between the two helices is really strange. Normally, an interface between two alpha helices in a structure like this will involve only hydrophobic residues, which will form the interface and escape from aqueous solvent. What we have is part of the interface built up by a chain of hydrophilic residues, polar residues, forming a whole chain of hydrogen bonds. So this is a very odd coiled structure. In fact, it's unique. It's the first time it's been described. But when we went on to solve more of these structures, it got even more complicated, because what we discovered was, in fact, that these alpha helices can have both arrangements. They can be both parallel or anti-parallel. And they use different interfaces to form parallel interface, different residues to form the parallel interface that they do form the anti-parallel interface. So the anti-parallel interface is the strange interface with the chain of polar residues. The parallel interface, when the helices are running in the same direction, is an absolutely normal looking coil with hydrophobic residues at the interface. So we were interested in knowing, well, in solution, is it possible for any one of these structures to form both orientations, or are the differences between one red septin and another? Some are parallel, some are anti-parallel, or can all of them be both parallel and anti-parallel? And what we discovered, looking in solution now, using NMR, and for this, once again, we needed access to big infrastructure, so we measured data both in Regensburg and in Florence, because we didn't have our access to high field spectrometers in São Carlos. And to cut a long story short, what this shows is that all of the sequences are able to form both arrangements. So they are both metastable. Neither the parallel nor the anti-parallel arrangement are particularly stable. And even when we looked at this theoretically, the collaboration of Ingemar and André, using a version of the Rosetta program, what he was able to identify was that two low energy structures represent, this is 50,000 different models that he's produced here. And the red ones correspond to the lowest energy structures. And you can see they come in two groups, and that basically corresponds to the parallel and anti-parallel arrangement. In other words, what that's saying is that the energies of both these two arrangements appear to be very much equivalent. So we can imagine a dynamic mechanism whereby these helices can be formed both parallel and anti-parallel arrangements. Indeed, these are just a superposition of Ingemar's predicted structures in yellow, and our experimental structures in blue, and you can see how good the prediction is. Even in the case of the anti-parallel arrangement, it correctly predicts the presence of the hydrophilic residues at the interface, and we have to remember that that had never been seen before in a coiled structure. So what does all this mean? What we suggest is that when a filament is forming, here you're seeing two paired filaments, but just look at the bottom filament. When a filament is forming, this red interface must come into existence. And for that, we expect to find parallel arrangement. But when these filaments start bundling up into higher order structures and sitting against membranes, then we can see that these helices could rotate by 90 degrees. They could pair up now in an anti-parallel arrangement with a helix coming from a neighboring filament, such that this lattice of filaments can extend in principle infinitely and sit on a membrane, which is here represented in gray, using the polybasic region, which I mentioned at the beginning, which is known to be important for association with membranes. Okay, and we personally think that the hydrophilic side of the interface here shown in green is probably stabilized, which you wouldn't expect in an aqueous environment. It's probably stabilized by the low dielectric constant when you get close to the membrane. Okay, so a lot of what we've been trying to do, as it were, over the last 10, 15 years is to what I call assemble a molecular jigsaw. In other words, to try and work out how it is that spontaneously, intracellularly, each septin knows its rightful position along the filament. So we saw, for example, in the case of the hexamer, that the yellow septins are at the center, then the blue, and then the red ones are at the end. And this is important for the way in which the filaments polymerize. And yet it's not a trivial problem to understand the subtle molecular recognition which must go on for each of the individual septins to know its rightful position along the filament. So a lot of what we've been doing is trying to assemble this molecular jigsaw. So basically, how does each one know its rightful position in this particular case, in the optima, with the red septins on the end, then the blue, then the yellow, and the green septins at the center? Well, as I mentioned, we have been spectacularly unsuccessful in crystallizing either hexamers or optimers, probably because these are long, thin particles, flexible. They don't crystallize very readily. So we have had to take an approach where we've divided this up into smaller parts. But before I go on to that, let me just emphasize the fact that it really is not a trivial thing to understand how this spontaneous assembly occurs. And the way I'll do that is just by comparing these different structures here. So at the top, you have the hetero-oligomeric structure with its blue, red, and yellow, which I showed at the beginning. But below it, you have an example from each one of the four groups where we've only crystallized a single septin. And yet, when you look in the crystal, expand the lattice, apply crystallographic symmetry such that you can look at the filament which forms within the crystal, you see that they all basically look the same as the original hetero-oligomeric structure at the top. So if I crystallize just the red septin, what I get is a structure which looks very similar to the hetero-oligomer at the top. If I do it with the blue, the yellow, the green, they all look basically the same. In other words, they all form these two kinds of interfaces, one on each side of each monomer, the G on one side and the NC on the other side. So for example, a blue septin in a proper hexamer-based filament, a blue septin would be sandwiched between a yellow one on the left and a red one on the right, form an NC interface with the yellow and a G interface with the red. But if I look at it down here on its own, when it has nobody better to pair up with, it just pairs up with itself. So one blue septin pairs up with another blue septin to form an effectively identical NC interface on the left, and with another blue septin to form an effectively identical G interface on the right. So when all of these things are being co-expressed simultaneously, being produced simultaneously within a cell, they've got to work out where they've got to go. And so that must be due to the kinds of interactions that we observe at each one of these interfaces. But this must be quite subtle given that they're capable of forming the same interfaces with themselves in the absence of a correct binding partner. So this is really molecular recognition, at least from a protein crystallographer's point of view, in a very subtle sense of the term. So how does each septin know its rightful position? And so we've taken the divide and conquer approach, given that we've been unsuccessfully crystallizing whole hexamers or octamers. And so, for example, we can crystallize a red with a blue, so they form a G interface. And we've also been able to crystallize 7 with 7, which forms a G interface in a hexamer, or yellow with green, which is a G interface in an octamer. We solved all of these structures, and if you take the structures apart and then look at the part of each monomer which is involved in the contact, so you imagine that we pulled the dimer apart, the complex apart, and opened it like a book, then what you're seeing here in red and in blue are the residues which basically make contact across the interface. And the first thing you can see is that it doesn't matter which blue septin we use. We've done it with 11, 8, and 6. They're all blue septins. When they pair up with a red septin, effectively the same contacts are made. And these are rather different when you look at yellow with yellow or yellow with green. So yes, it does seem Kinoshitas Rule makes some kind of sense. You can substitute a blue septin with another blue septin simply because it provides the same contacts at the interface as one of the other blue septins. So if we go to look specifically at that interface formed between the red septin and the blue septin, okay, they form a G interface. Red never forms a G interface with yellow or green or with itself, and blue never forms a G interface with itself or with green or with yellow. It's always red with blue. So where does this molecular recognition come from? So what we've observed, going back to sort of jigsaw puzzle analogy here, is that there's a special residue in the red septin, it's an alanine, which pokes into a special pocket on the blue septin, which is formed by these 4 residues here. It turns out that only the red septins have this residue, and only the blue septins have these 2 residues here. So this particular combination, which forms this contact, can only exist between red and blue. And on the same thing, same thing happens using a different residue at the bottom. There is a blue specific residue, which is a histidine shown here, which only exists in the blue septins, and it forms a contact within a cavity which is formed by these red specific residues over here. So despite the fact that these are all similar folds, similar structures, 3-dimensional structures, there are subtleties which mean that it's possible to for selectivity to be built into this system. And that's how spontaneous assembly is able to occur. Turns out in the blue septin, something else is also true, and that is that there's a whole chain of interactions which lead to this histidine residue, and they start at the gamma phosphate on GTP. I told you that these molecules bind GTP, which can be hydrolyzed to GDP. And this is the last phosphate. This is the phosphate which is lost when hydrolysis occurs. But the septin 6 group, or the blue septins rather, are unable to hydrolyze the GTP to GDP. So this phosphate is always present and is part of this network of interactions which builds selectivity into this interface. So by taking all of these things apart and solving dimeric structures, we can then reassemble a pseudo hexamer at pseudo high resolution. Remember, the original hexamer was only at 4 angstroms resolution, with large chunks of it missing. Lots of the loops and the side chains were all missing. But what we can do here is take our high resolution structure of individual pairs and just reassemble the thing, so that we can have a pseudo high resolution structure of a hexamer, which would look something like this, which is now complete. But of course, it would be really nice if we could look at the hexamer directly, rather than having to reassemble it from its parts. And because, as I say, we've been unsuccessful in trying to crystallize hexamers or octamers, we've had to start getting into cryoelectron microscopy as an alternative structural approach for looking at the hexamers. So once again, we've been going after infrastructure all over the world. We've been to Leeds, we've been to Stanford to try and get access to Titan Cryos microscopes. But in fact, we now have one in Brazil at the synchrotron laboratory itself in Campinas. And the data which I'm going to show you is actually all being collected here in Brazil. So this is a raw image with these little sort of little rods are hexamers, which you're seeing in the raw image. These can be classified into different classes and then used to produce a three-dimensional reconstructed image for the hexamer. So this is the hexamer here at the bottom as a surface representation. Its average resolution is 3.6 angstroms, but its local resolution varies quite considerably. And in this particular dark blue region of the structure, the map looks very good indeed, and it's better than 3 angstrom map. If you were to compare it with what a 3 angstrom map looks like from an X-ray diffraction experiment. Okay, so what that means is we're now able to look at the hexamer directly. And what we observe is if we look at this particular interface, now this is one which I haven't been talking about much up until now, it's the interface from between yellow and blue. It's one of those interfaces which I call an NC interface. And if you look down on the figure on the left at the bottom, you will see in this surface representation of the structure, there's a huge cavity between these two monomers, between the blue on the right and the yellow on the left. So there's a big sort of defect at the interface. It's very different from what I was just showing you before at the interface between the blue and the red. The blue and the red septins seem to fit together like a nice jigsaw with these very specific interactions which guarantee that it's only blue with red and not any other combination. But at this interface between yellow and blue, there's this huge cavity. It looks like there's a defect at the interface. It's about 4,000 cubic angstroms. And at the bottom of the cavity, down here, on the right, you can see this looking from the top down. But what we see are 2 alpha helices, one from each subunit, which form the sort of floor of this cavity. Now, these alpha helices are very important functionally because we know that that is where we have this cluster of basic charges. So what's called the polybasic region, that is all concentrated within these alpha helices. It's also known that this polybasic region is important for interacting with membranes. So just going back to the very beginning, septins only function because they can self-polymerize, bundle, and interact with membranes, and therefore deform membranes as in during cell division. So the interaction with membranes is fundamental, and it's known to be dependent on the presence of this polybasic region, which basically corresponds to these alpha helices here. Okay, but what's strange about the structure when you look at it in detail, and here we're looking at it basically the same thing here, but a little bit more detail, and more the yellow alpha helix I've now colored green, just to confuse everybody, no, just to highlight it. And here you can see you have an arginine, a lysine, another arginine, another lysine. This is the polybasic region. But what's curious about it, given that we know that this region supposedly interacts with membranes, is that it's completely hidden. It's not exposed. There is no way a biological membrane can interact with this helix the way it is tucked away, hidden within the NC interface, between the interface between blue and yellow, forming the base of that cavity. So that doesn't seem to make much sense. I mean, we know from other experiments that this region is important for interacting with membranes, but how on earth can it interact with membranes given that all of its positive charge are taken up with salt bridges within the interface? It looks as though it's been deliberately put there to be hidden away and in a confirmation for not interacting with membranes. So if this thing really ever does interact with membranes, something's going to have to change. So that interface between yellow and blue would have to squeeze and expel the helix such that it exposes its basic charge. Well, we've never seen that happening in the case of yellow and blue, but we have seen it happening in our structures for the green septins. So what you're seeing here is a little morphing going on. This is a structure which only has green septins, okay? And when we have the structure bound to GTP, this interface is closed. When we have it bound to GDP, the interface is open. This is a big change. It's about 8 angstrom movement. It's big enough for when it closes for there to be insufficient space at the interface for the polybasic region. So it must get kicked out due to steric hindrance. But when the interface opens, that creates the cavity big enough for that alpha helix to be hidden away and safely keeping its positively charges out of trouble. So what that seems to suggest is that this whole filament should be able to shrink and expand as a function of whether the septin's bound to GTP or GDP. In other words, the hydrolysis of GTP to produce GDP will expose the, or will alter the opening and closing of the interface such that binding to membranes can be controlled. It's really quite interesting what happens when the interface opens and closes. This is on the left is the open interface. There's a whole bunch of salt bridges which are formed in the open interface. When the interface closes, those same residues are all of a sudden become involved in a completely new set of salt bridges, and they involve this region here shown in yellow, which is called a polyacidic region, which is partly involved in interacting with the polybasic region of the alpha helix itself. Okay, so for the most part, our structures don't have that alpha helix. This is why it was new to us when we looked at the Cryo-EM structure, because in the Cryo-EM structure, that alpha helix is present. But in the crystallographic structures, we normally remove it because it helps crystallization. So that meant we had relatively little information about this alpha helix, which is why the Cryo-EM was so important. But we do have one structure where the interface is closed and the alpha helix is present, and it's the green structure shown here. And indeed, the alpha helix is sticking out of the interface. There is simply not enough room at the interface for this alpha helix to be hidden away as it is in the red structure here, which is the structure where the interface is open. So that's probably why the cavity exists. The cavity exists such that this interface can open and close, and that this, as we predict, is important for how the filament interacts with the membrane. So in the presence of GTP, positive charges are exposed that can interact with negatively charged membranes. Hydrolyze GTP to GDP, that opens the interface, and the positive charges can be safely hidden away out of harm's way. So that would be a mechanism by which you could control whether how the filament interacts with and comes off from membranes. Well, this slide is a little bit redundant, really. It's just talking about this polyacidic region here shown in red, which when the cavity is open, it interacts with the basic residues on the little alpha helix. And when the cavity closes, then the polybasic region on the alpha helix gets kicked out so that it can interact with membranes, and the negative charge on the polyacidic region is stabilized by a second region, also rich in basic amino acid residues. So that's the basic message for how we think then the hydrolysis of GTP is coupled to the way in which the filament interacts with membranes. Now, the way in which septin filaments interact with membranes is curious, because there's a lot of data in the literature suggesting that septins are capable of recognizing curvature in membranes. They may induce curvature, but they also appear to be able to simply recognize curvature. And if we just take all of our different crystal structures and use them to assemble different possible octamers, we can see that there's quite a bit of flexibility in the octamer that's shown here on the right. So these are reassembled octamers. There's no structure available, neither from X-ray diffraction nor from Cryo-EM for a full octamer. These have been reassembled from their parts, from different structures. And you can see there's quite a bit of bending going on to the particle. Also, that central interface can open and close, which is what I've just been talking about. But what about experimentally? What can we see in terms of particle flexibility experimentally, not from a crystallographic study, but when we're looking at single particles with Cryo-EM, then it is possible for us to see a degree of variation within the particle. So here's a little movie where we're just morphing from two extremes where you can see that the particles are not all straight. There's bending going on, it's what we call side-to-side bending, which is more predominant than the up and down bending. And here you can see that it's mostly the central interface here where that movement is occurring. And we would expect, once we have an octamer with two green septins in the center, that bending will be even more noticeable. And if the particle bends such that it follows the curvature of the membrane down at the bottom, then that will point the opening to that cavity where the polybasic helix is, that points towards the surface of the membrane itself. So what could be the importance of this filament bending? Well, since we have now established that the red septins are at the ends of both the hexamers and the octamers, then, as I've already mentioned, it should be possible, and this has been experimentally demonstrated, that you can form mixed filaments which could be composed anywhere between 100% hexamers all the way to 100% octamers and any combination in between. So what that means is that the red, blue, and the yellow will always be present in stoichiometric quantities, but the green septin depends on the dose of octamers that you have within your filament. Since the green septin appears to present these unusual properties of flexibility and being able to open and close its interface, you could dramatically change the properties, particularly in terms of membrane association of a filament, by controlling the dose of octamers. So a filament composed just of hexamers will be a filament with one kind of property, group of properties, whereas one which has a higher dosage of octamers could have completely different properties in terms of its flexibility, membrane association, detection of membrane curvature, etc., etc. This is really, I'm really now getting into the future because we know very little about this. This is still really speculative, but we are currently trying to work on the structure using Cryo-EM of an octameric particle, well, both from a human septins and from a model organism called Pseudohigh intestinalis, which is better behaved from the point of view of the octamers we can produce. Okay, so it's time for me to wind up. And what have we learned and how have we learned it? Well, with respect to septins, we've started to learn, I think, quite a lot. So we've learned the importance of parallelism and anti-parallelism of the coils on the red septin, important for establishing the formation of a filament when they're parallel and cross bridges when they're anti-parallel. And of course, if you don't have this part of the structure, if you don't have this alpha helix because it's been cleaved, in the case, for example, of the Zika protease, then you will be unable to form a stable interface necessary for polymerization. We've learned something about what happens, or about why we have selectivity, or where selectivity comes from at the interface between red and blue, and the importance of the fact that the blue septins are catalytically inactive. We've learned something about the importance of opening and closing of interfaces, particularly the green interface, where we see this in our crystal structures. Some things which I haven't mentioned too much about, but the fact that the GTP hydrolysis must occur at a G interface, but the structural change occurs at an NC interface, means that there must be information transfer from neighboring interfaces along a filament. So we learned quite a bit about this, how this fascinating oligomeric structure self-assembles and how that's probably related both to its normal physiological roles and also to how it might be involved in disease, particularly in the case of Zika virus infection leading to microcephaly, although of course most of the details are still unknown. But we've also learned one or two other things, I think. And so I'll just put 3 bullets here to wind up with. So integration at the national and regional levels means that we can advance more quickly. And it's so encouraging to see the formation of the African Crystallographic Association, the initiative to build a synchrotron for the entire continent. And clearly, I think this is almost a no-brainer, really. We do have to collaborate, especially when there's relatively few of us, where the critical mass is still to be formed. Access to international infrastructure is essential for maintaining competitivity. That has been our experience. We go all over the world, wherever we need to go, or we send our samples, do whatever kind of experiment we need to be able to do in order to try and keep ourselves competitive. And in the case of Brazil, I would say that building a synchrotron was a catalyst for growing structural biology in Brazil. There are many good reasons for building a synchrotron, but one of them is the fact that it's a huge stimulus for growing a local community, and not obviously just structural biology, but many communities which are synchrotron radiation users. So just finally, to thank all of my friends and colleagues and postdocs and students and collaborators. This is our current group in São Carlos. Many former group members have also contributed the work which I've described. And of course, it's them who does most of the work. And I thank you for your attention. It's been a pleasure.
Thank you for an excellent talk. That was very inspiring, particularly seeing how much the community in Brazil has grown since you've been involved, and I think that that's very much an inspiration for us in Africa to go ahead with everything that we've been doing, and as you said, with the AFCA and so on. So for me, it's very exciting to see what is possible in the future. So I would like to open the floor up to questions. I haven't seen anything in the chat, but please feel free to type something. I see we have a question from Sylvia. Please go ahead and unmute yourself and ask away.
Mm-hmm. Hi, Richard. First of all, congratulations. This is it's a very difficult, subject with a lot of complication, and you're doing an excellent job in, using color and diagrams to explain it. I was wondering whether these proteins are actually present also in archaea, whether you can learn anything in terms of redundancy and evolution of these.
This is a very good question. They're not present in archaea or bacteria, and in plants, they're also absent. So they're fairly widespread, but there are some notable exceptions. But what's particularly curious is there are species which have a large amount of redundancy, such as humans, for example, 13 different septins. And if a particular color can be substituted by another color, by another one of the same color, you might ask, well, so why do you need both of them? Why do you have 5 blue septins? I mean, if they can all be substituted amongst themselves, I mean, what difference does that make? Why do we need so much variation? I suspect that it comes from the fact that the N-terminal domain, which I didn't say anything about, is very variable from one septin to another. So different combinations of septin will bring together different N-terminal domains, which vary considerably. And these are known to be important for recruiting binding partners to the septin scaffold. So depending on whether the septins are being involved in cell division, in exocytosis, in vesicle trafficking, there's a whole myriad of different functions which have been attributed to septins. And so depending on what function it is that you require, that probably requires a different combination of N-terminal domains, which means that you must bring the right septins of each color into the right position. But other species don't have such redundancy. So Schistosoma mansoni, for example, basically has 1 copy of each type, and it doesn't even have a green septin. Siona intestinalis has 1 copy of each one. The fruit fly has 2 copies of some and 1 of another. And we really don't understand very well why this sort of intrinsic redundancy is necessary in some cases and not in others. But it would appear that as there's been an expansion of the genome, new functions have appeared for different septins or different combinations of septins. But archaea and bacteria don't have septins, nor do plants. Thank you for the question.
Fascinating. Do we have any more questions? Well, maybe I can ask one quickly. So what do you think the possibility is that we'll ever be able to observe octamers? I mean, you said that you haven't been able to. Do you think you'll be able to do it?
I think we will, yes. I don't think that that's a fundamental issue. I think with cryo-EM, we should be able to do that. Yeah. One of the great advantages of cryo-EM is that if you have enough particles, enough computing power, then you can separate out different conformations from within the particle data set. This is not trivial, but what it does mean is that if you do have these long stringy octamers bending to different extents, then if you try and use them all simultaneously for reconstruction, obviously you lose resolution. But if you can identify only particles with a particular kind of bending and separate them out from the others, then you should be able to regain high resolution. I'm not really a specialist, but I'm optimistic with the way that Cryo-EM has proceeded over the last few years. I'm pretty optimistic that we'll be able to use Cryo-EM for an octameric structure fairly soon. The question, an interesting question, I think, is how much extra information does that bring you compared with just looking at the individual pairs? And I think that's not really clear. I mean, we may have to sort of work very hard to get an octameric structure. And then when we look at it, we realize that, well, it's not actually telling us anything new. We knew all of this just by looking at the individual interfaces separately. But that's not going to put us off trying to do it, because normally you get a surprise. So we're still trying to solve an optimal at a decent
Wonderful. That sounds great. Well, I wish you good luck with that. And I don't see any more hands. And I think we've reached the end of our session. Oh, wait. Sorry, Sylvia, I beg your pardon. You have another question.
I don't want to take too much time, but I'm also intrigued by the role of the GTP, and does it, are these very good at hydrolyzing GTP? No. Do they need any help, any gap again?
Oh,
how does it?
Right, good question. Yes, no one has ever successfully identified a gap or a GEF for a septin. It may well be that they aid one another because the GTP binding site is at the G interface, and there are known examples where a protein, a small GTPase, can be its own activating protein. But in the case of septins, we have no specific information about that. But no, they're pretty lousy catalysts. Like most GTPases, they are catalytic rate is very slow. And we're still not entirely sure of how important or how viable exchange of the nucleotide is once the filament has formed. How readily a GDP, for example, can form off a filament for another GTP to bind. These are still very open questions. Yeah.
Thank you.
Does that finish answer your question, Sylvia? Fantastic. Okay. Any more questions? No, it doesn't look like it. So before we thank Richard again, I think we should take a photograph. Delia, are you able to do so?
I am indeed.
Fantastic. So if everyone would switch on their cameras, then Delia will take photo. Delia, will you count us in?
I will. I'm just waiting for a few more cameras to go on, and just to remind everyone that I'm going to have to click through three screens to get you all. Fantastic. So you have to smile for a long time because you don't know which of my screens you're on. Okay, so I'm going to do the first one of my screens now. So I'm going to ask for a big smile on 3, 2, 1. That's the first one. And this is the second one. 3, 2, 1. And then just one more. 3, 2, 1. Well done, folks. Good smiles. Thank you very much.
Thank you.
Fantastic. Thank you so much. And thank you, Richard, for a really, really interesting talk. I'm sure everybody is equally as inspired and excited by the work that you're doing as I am. So if everybody could just use your reactions to do a quick clap or switch on your speaker and clap your hands, then we can thank Richard. And thank everybody for their attention. Delia, can I pass over to you for any final comments before the end of the day?
Sure. It's not actually the end of the day for many people. It is for ePCCr. So ePCCr people, thank you very much. We'll see you again tomorrow. But otherwise, there are both AfLS and AfPS sessions starting at 5:15. So we'll see you guys there. Thanks very much, everyone.